Hi, welcome back to the ESS 330 lecture on ecosystem modeling and the Century ecosystem model. This is part 2 of 4. In part 1, we looked at some common terms in ecosystem modeling and some simple box and arrow diagrams of models. Part 2, we're going to focus a lot more on the Century and the Descent ecosystem models. And also we'll discuss a little bit about some other types of soil organic matter models that are being currently developed. So the descent model has a lot of pools and fluxes, boxes and arrows like you've seen in some of the examples that I showed you in part 1. But it's a little bit more detailed than the simple ecosystem model that I showed you. The plant components in the descent model are not just a single big box of carbon, but are divided into boxes of leaves and fine roots, and if we're growing trees, branches, large wood, and coarse roots. And when plants die, they become litter, and in the descent model, that litter is divided into structural and metabolic litter, and I'm going to explain all these terms in the upcoming slides. This is just an overview. Then when plant material decomposes, it releases CO2 through heterotrophic respiration, and then some of that plant material becomes soil organic matter. And soil organic matter in the descent model is represented as 3 different types based on how long they persist in the soil in general. So there's the active, slow, and passive pools. There's also soil layers that in a soil temperature and soil moisture model in the descent model, and there's also inorganic nitrogen in the form of nitrate and ammonium that also occurs in multiple layers in the soil. And in this particular diagram, the solid arrows are fluxes from one pool to the next, whereas the dotted arrows or the dotted lines are meant to communicate some sort of dependence on another pool. So for instance, you'll see that an arrow from the soil temperature and moisture is pointing to the death of the plants, the plant components. So in that sense, the plants could die at a quicker rate if the soil temperatures are too hot or too dry, or the soil moisture is too dry. And the soil temperature and moisture also affects the decomposition rates of soil organic matter, as we already discussed. And one of the main purposes of developing this layered inorganic nitrogen pools in descent is so that descent can simulate nitrification and denitrification processes, and those produce nitrogen gases. In particular, it's the N2O gas, the nitrous oxide, that we're interested in from a standpoint of greenhouse gas balances. The model also simulates methane uptake and methane emissions, and I'll mention that a little bit later. So what is the Century model? Well, it's an ecosystem model that simulates carbon and nutrient cycling between plant pools and soil organic matter. It always runs with both carbon and nitrogen, but you can also run the model with phosphorus and potassium and sulfur if you wish, or you could just focus on CNN dynamics alone. This model was developed at the Natural Resource Ecology Laboratory in the 1980s. It is called Century because it simulates soil organic matter formation over centuries. It runs at a monthly time step, and soil organic matter decomposition is first-order linear decay, like the examples I showed you in part 1. It can simulate a variety of ecosystems, including grasslands, savannas, forests, and agroecosystems. And one of the strengths of the Century model is that there are these schedule files where a user of the model can specify what plants are grown in what years, when the plants start growing, and if it's a agricultural system, when the plants are harvested, how much is harvested, if the soil is tilled, if there's any kind of ecosystem disturbances such as fire or wind damage. There's just an unlimited number of scenarios that you can create with these schedule files to tell the ecosystem model more about the kind of system you want to simulate, and to do experiments with the model too. This model is written in Fortran 77, and I bet there's many of you have never even heard of Fortran. It's a language that's been around for a long time, but it still works, and still works well. And the Century model is about 19,000 lines of source code and comments, which is a little bit bigger than some of the R scripts that you've written for this course. There's a really interesting history of ecosystem model development, in particular because a lot of ecosystem model development happened at Colorado State University. So back in the 1960s, from like about 68 to 78, the Natural Resource Ecology Laboratory, NREL, received substantial funding to manage the U.S. International Biome Grassland Program, the IBP Grassland Program, and to develop an ecosystem model that was capable of simulating grazing and climate impacts on grassland ecosystems. This had never really been done before. And the first model that came out of this effort was known as the ELM model. And the ELM was developed by a team of scientists with a wide variety of backgrounds, including mathematics, meteorology, soil, carbon and nutrient cycling, plant production, animal production, and statistics. And it was challenging because there were a lot of people with diverse backgrounds working together, and biologists at that point didn't have very much experience in model developing. Most of model development had occurred up to that point was by physicists and mathematicians, so there was no references for biologists to go to learn about how to do ecosystem modeling. It was a pretty new practice, a pretty new science. And you can imagine, or maybe I hope you can imagine, that how poor computation efficiency was, 50 years ago relative today. I mean, look at the computer at the lower right of your screen. That was probably the best that they had to work with at the time, and sometimes they didn't even have a computer at all. They had to use like punch cards that didn't have a screen. Because of all of these limitations, and because everybody wanted to represent all the processes that were happening in the ecosystem simultaneously, it became a very challenging project, and the ELM model was too complex and it did not work very well. So that's when Century came along. The biologists took a step back and thought about what were the most important things to simulate in order to be able to represent grassland ecology well. And so Century is a simplification of ELM. The descent model. The descent model came about 10 or 15 years after the Century model, and it's just a daily time step version of the Century model. So Century was a monthly time step, and descent went to a daily time step. And it was first developed at NREL in 1994, and I was part of that effort. And the daily time step was needed, and a more detailed soil water model and more detailed layered inorganic nitrogen were necessary to be able to simulate nitrification and denitrification, because it was becoming increasingly important to be able to simulate nitrous oxide emissions as well as the carbon cycle. The mineral nitrogen pool, which was just one inorganic nitrogen pool, was split into ammonium and nitrate species. And development is ongoing these days. Recent development has included being able to simulate wetland systems and methane production or methanogenesis, and developing new plant types like fruit trees and large perennial biofuels grasses like miscanthus and switchgrass. And like Sentry, DayCent's used by researchers around the world, and there's hundreds of publications that include Sentry and DayCent applications. And many versions exist because there hasn't just been one research group using DayCent. Multiple research groups use it. They modify the code to do something specific to meet the requirements of their research. And so lots of different versions of DayCent have been developed, but the things I'm going to talk to you today about are pretty common to all versions of DayCent, no matter which version if you encounter DayCent, this would be common to all those versions. It's also written in Fortran 77, but also Fortran 90 and the C programming language, and it's over twice as many lines of code as the Sentry model, about 44,000. And DayCent is the core of some other greenhouse gas inventory tools, including Comet Farm and Comet Planner. And it's also the basis of the US EPA National Greenhouse Gas Inventory. For example, the Comet Farm tool. It's a web-based, GIS-based tool for a farmer or a rancher to evaluate how the management practices on that land will affect the carbon and greenhouse gas balances. So the farmer could enter all kinds of information about how his crops are managed, what types of soils are in his fields, and underlying the calculations is the DayCent model, which would compute the greenhouse gas budget for each of those practices. So what kind of model is DayCent? You may have seen in some previous lectures in this course that there's different ways of categorizing models. And models can have a combination of these characteristics, but there was the statistical or theoretical or process-based model, and DayCent is a theoretical or process-based model. It's based on laws of nature. It's not just based on data relationships and linear regressions. There's deterministic versus stochastic models, and DayCent is a deterministic model because the same input produces the same output. There's no stochasticity, there's no randomness in the DayCent model. Static versus dynamic. DayCent is a dynamic model because it changes over time. It runs at a daily time step, can run for thousands and thousands of years if you wish. It's not just a one-time calculation. Phenomenological or process-based. This is a little trickier to describe, but again, DayCent is a process-based model. It's modeling the processes. It's modeling the plant growth each day, and the amount of biomass that ends up at the end of the growing season is accumulation of all the daily accumulation of biomass that happened over the growing season. There's no prescribed pattern of how a crop or a grass is supposed to grow over a growing season. And if you go back to that starling murmuration example, a pattern would be looking at that flock flying as one big giant organism and trying to simulate the patterns of the flock as a whole, whereas a process-based model would be simulating the individuals in that flock and what the individuals are doing and how that pattern results from all of the individuals in the model. This can be a little fuzzy sometimes to distinguish between phenomenological and process-based. You won't be running the DayCent model, so you don't really have to understand all the details of how the whole system is set up. But I have run into a few people when I co-taught ESS 330 a few years ago that had actually used the DayCent model. And you know, it's possible if you stay in ecology, even if you don't become an ecosystem modeler, you may still hear of the DayCent or Sentry models. The Sentry or the DayCent model is an executable program. It has input files and output files. So all the arrows going into the DayCent models are meant to be input files. All the arrows coming out are its output files. Its input files include a schedule file, like I was telling you about just a little bit ago, a weather file that describes daily precipitation and minimum and maximum air temperature, all kinds of different parameter files to describe the types of crops, the types of trees, the cultivation practices, the fertilizer practices, harvest practices that the model simulates. And the model produces spreadsheets as an output, CSV files or other types of text files, and a binary file, which can be translated into a text file. We'll talk about boxes and arrows. This is the representation of the carbon cycle in the descent model. And there's plant pools, and there's soil pools, and there's plant litter pools all in this. You certainly don't have to memorize this. I certainly don't even have it memorized after 28 years of working with the descent model. But just you can think of, when I showed you those 2 pool soil organic matter models in part 1, you could pull just about any 2 boxes that have an arrow between them and think of them as like the 2 pools that I showed you in that simpler diagram, and think of the processes that would affect the transfer of carbon from one pool to the next. So the descent model has herbaceous pools above ground. Pools can die and become standing biomass, and both plants die. When they die and fall to the ground, they become litter, and litter becomes soil organic matter. And the left side of the diagram is mostly the above ground component, above the mineral soil, and this right part of the diagram is mostly meant to represent the below ground portion of the model. In descent, all carbon is either expressed, if it's a pool, it's expressed as grams of carbon per meter squared. And if it's a flux, it would be grams of carbon per meter squared per day. So it's just carbon. Even though it technically simulates respiration, which is CO2, it's just always looking at the carbon portion of pools and fluxes. Since organic matter includes both carbon and nitrogen, then the nitrogen cycle, which is represented here, looks a lot like the carbon cycle, except that it's even more complex. Again, like for every carbon pool there is in the plants, in the soil, and the litter, there's a corresponding nitrogen pool. And the left side of the diagram is the above ground processes. The middle part of the diagram is the soil processes. And what's different about the nitrogen cycle is not only do you have to consider the organic nitrogen that's cycling through the system, but also its interaction with the inorganic nitrogen pools and fluxes. Because plants take up inorganic nitrogen, soils, when they decompose, release inorganic nitrogen, so it adds to the complexity of the nitrogen cycle. Okay, I'm going to go into more detail about some of the pools and descent that you've heard me talk about. Plant litter. So when a plant first dies and that material falls to the ground, it hasn't decomposed yet, it's still litter. And there are 2 types of litter in the descent model. There's the metabolic pool and there's a structural pool. And the metabolic pool is called labile or easily decomposed. And in the model, its variable name is METABC. And there's an above and a below ground component of the metabolic litter. The structural litter is much more resistant to decay. The structural litter contains all the plant lignin, the cellulose, the hemicellulose. So if you think of some dead corn stalks, a lot of that material is structural material. It's supporting the plant leaves. It's supportive structure in the plant. And so, and wood too also has cellulose and hemicellulose. So those decay slower. So the way that descent determines if you take plant litter, how do you determine what's metabolic and how do you determine what's structural? And that's based on the lignin to nitrogen ratio. So if a plant is very low in lignin and high in nitrogen, that's a smaller ratio, and that would be the metabolic component of that dead plant litter. Versus if plant litter has a lot of lignin and not so much nitrogen, it has a high lignin to N ratio, then that material would be referred to as structural material. Okay, soil organic matter representation. I mentioned these before: active, slow, and passive. They're based on the mean age of the pool, or you can also think of it as the turnover time, which is just 1 over age, or the turnover rate. So the active pool, in the model, it's abbreviated SOM1C, and there's a 1 and 2 component for above and below ground. The active is considered the live microbe pools and their byproducts, and it is about 2 or 3% of the total soil organic matter, and has a lifetime of about half a year to a year. The slow pool is chemically resistant to decomposition, and it is about 49% of the total soil organic matter, roughly. It has a lifetime of 10 to 50 years. The passive pool is physically protected from getting attacked by microbes, or it's chemically inert like charcoal. So soil organic matter absorbs to clay particles. It becomes protected so that microbes can't access that carbon as easily, and so it's slower to decompose, and it's called protected, physically protected. It has a much longer life than the other pools, 100 to 1,000 plus years. So looking at this red box here, there's two rectangles. The upper rectangle is meant to represent above the mineral soil horizon, and the bigger rectangle is meant to represent below the mineral soil horizon, or mixed in with the mineral soil. So the active pool and the slow pools have above and below ground components, whereas the passive pool only has a below ground component, because for carbon to become protected, it has to interact with clay, and we don't think of clay as being above the mineral soil horizon. Clay minerals are within the soil itself. And the plant litter is the carbon that's not decomposed yet, that will when it becomes decomposed, it goes into one of the soil organic matter pools. And above the mineral horizon, that plant litter would come from falling leaves or falling trees, and below the mineral horizon within the soil, that plant litter would come from plant roots that die. So back to our first order linear decay model, modified by environmental scalars. This is almost exactly what we saw in part 1, except there's a few more details here, they're more specific to the descent model. So we have one pool, soil organic matter pool A, I'm just using a generic name, that decomposes and sends carbon to soil organic matter pool B. There's a transformation. So maybe part of it's decomposed in pool A, and then the undecomposed or transformed material goes to pool B. So there's input to pool A, which is plant litter, and then soil organic matter pool A decomposes, and that decomposed material is what I'm calling C flow here. C flow is equal to the amount of carbon in pool A times a maximum decay rate, Kmax, times a temperature effect on decomposition, 0 to 1 multiplier, times a soil moisture effect on decomposition, another 0 to 1 multiplier. And this is new, a pH effect on decomposition. So really acid soils would decompose slower than soils with a more neutral pH, and a cultivation effect. And so you've seen examples of F of T and F of W, the soil temperature and moisture modifiers on the decomposition rate. We haven't really talked about how decomposition is affected by how much the soil is disturbed by cultivation. So this is not a 0 to 1 multiplier. This is actually a multiplier that's 1 or greater. So when there's no cultivation at all, for instance, if you're in a grassland that's not being cultivated, then the cultivation effect is 1. That just means there's no effect of cultivation on the rate of decay. But if you have a agricultural field, for instance, that's plowed up deeply by some sort of plow, it's chopped up into little bits, and it's highly disturbed, that's going to really accelerate the rate of decay of that pool. So a very highly disturbed soil might have a decomposition cultivation factor of 5, 10 or so. Whereas if you have a conservation tillage practice or a no-till practice, that number will be much closer to 1. I just pulled a small piece out of that much larger diagram of the century carbon cycle, and this is just looking at the metabolic and structural litter in the soil and 3 of the soil organic matter pools in the soil. So just wanted to kind of review some of the things that I've just talked about. The main thing to remember here is that these pools are based on their life, how long they persist on average. And the soil metabolic litter pool decays fast, where the structural litter pool decays slower. The active pool decays fast, the slow pool decays slow, and the passive pool decays extra slow. And the way that carbon gets into the passive pool is it gets sorbed to clay particles that carbon is protected against microbial attack. So that becomes protected. And the amount of protection increases with soil clay content, at least in the way the descent model works. Okay, let's talk a little bit about nitrogen. Mineralization and immobilization. Mineralization is the conversion of organic nitrogen to inorganic nitrogen, such as when soil organic matter decomposes. Soil organic matter has not just carbon in it, but has nitrogen in it, and when that soil organic matter decomposes, it releases the nitrogen, and that becomes inorganic nitrogen. And when microbes consume organic matter with a low carbon to nitrogen ratio, that means that the plant, the material is relatively rich in nitrogen, then they mineralize nitrogen, they release nitrogen as ammonium into the soil. And then that inorganic nitrogen becomes available to plants and can act sort of like a fertilizer. And disturbance will accelerate decomposition, and so if you're any kind of cultivation event that accelerates decomposition will also mineralize a lot of nitrogen. Immobilization is the opposite of mineralizations. Instead of microbes releasing nitrogen into the environment, they're extracting nitrogen from the environment during this process. So microbes consume organic matter with high C/N, which means it's poor in nitrogen. They have to pull organic nitrogen from the soil environment and to consume it to satisfy their needs for nitrogen. And the process of immobilization reduces the amount of inorganic N available to plants. So that's like maybe if you're a gardener, that you know that if you throw a bunch of wood chips on your garden, that when the microbes are decomposing those wood chips, because those wood chips are high in C and low in N, the microbes have to remove some of the inorganic N from the soil environment to satisfy their nitrogen needs in order to decompose the wood chips. So this is a simple way of showing you how descent represents nitrogen mineralization and mobilization. And so these pools in this, there's a soil organic matter pool A, and it has a carbon to nitrogen ratio C to N of A. And then there's a soil organic matter pool B, and it has its own carbon to nitrogen ratio B. Soil pool A is a source of carbon, and pool B is its destination. So as we saw before, when soil pool A decomposes, there's a flow of carbon out of it. Some of that carbon that flows out of pool A respires as heterotrophic respiration, and what's left goes into pool B. So that's the carbon part of it. But that carbon flow coming out of pool A also has a carbon-nitrogen ratio associated with it. So if you want to figure out how much nitrogen is in that carbon flow, you take the carbon flow and divide it by the carbon to nitrogen ratio of pool A. So N flow of 1 is the flow out of pool A, the carbon flow divided by the carbon to nitrogen ratio of that pool. And the way descent works, the way it figures out if nitrogen is immobilized or mineralized is based on the relative differences between the C to N of pool A and the C to N of pool B. So when pool A is nitrogen poor relative to pool B, or in other words, when pool A has a higher carbon to nitrogen ratio than pool B, then what happens when there's that flow of material from pool A to pool B, some inorganic N is going to get pulled into the flow before it goes into pool B. Conversely, when pool A is nitrogen rich relative to pool B, meaning that the C to N ratio of A is lower than the C to N ratio in pool B, then some of that nitrogen can be released when that soil organic matter flow is when that soil organic matter is flowing between the two pools. So that's how descent computes nitrogen immobilization and mineralization. So the first order linear decay method of modeling soil organic matter has been around for 40 years or more. And there's been a lot of new ideas that have come about better ways or maybe more realistic ways to model soil carbon dynamics. It doesn't mean that first order linear decay isn't useful. It's still very widely used. But there's other ways to do it. For instance, the newer models often have a live microbial biomass pool. So instead of an active pool, which is really just a static pool, it's supposed to represent microbes, but it doesn't really change, it doesn't grow and die like a microbe pool would. A microbe pool would be like an organism or a bunch of microbes that pool would get bigger if there was a lot of material input into the soil, or if the microbes were starved, then there might be death of microbes and a smaller microbial biomass. So instead of the rate of decomposition being controlled by a first order maximum decay rate, it's controlled by the amount of live microbial biomass in the model. And that's this top model. This on the right side, what's called the millennial model, is just a modeling paper I was part of where they were looking at different ways of representing soil organic matter decomposition. Another difference main difference between the descent model and some more contemporary models of soil organic matter is that the descent models pools, active, slow, and passive, are really based on how long they live in the soil, and they aren't necessarily directly measurable. To some degree they are, but they're not really. Like if you're familiar with any kind of fractionation techniques and measuring soil carbon and determining different components of soil organic matter, those don't work well for determining the active, slow, and the passive portions of soil organic matter. So one goal of more contemporary models is to represent models or represent pools such as POM, particulate organic matter, and mineral associated organic matter that are more easily directly measurable. So that is the end of part 2. So I will see you at part 3.